Okay, let's get started then. Thank you everyone for attending the session on processing and analysing data this morning. For our fourth talk this morning, we have Jeff Christiansen and Anifa Kanan. Jeff is a senior business analyst at the Australian National Data Service, ANS, and is responsible for liaison between ANS and the software development team at Monash. And Anifa is the program manager, software development at Monash eResearch Centre. The title of the talk this morning will be The Proteome Browser, a software resource for the Chromosome-Centric Human Proteome Project. Thank you very much.
Thanks, Bernie. This is a project, as Bernie mentioned, is based at Monash University and has been developed by the Monash University eResearch Centre. And it's a ANS-funded project in the applications program, and the aim of that program is to fund software resources that are going to bring together different data sources and different data types to develop tools to meet a research need. So, I should just say, so yes, as Bernie mentioned, the project's called the Proteome Browser, and this is a tool that's being developed for a chromosome-centric human proteome project. So, what's a proteome, you may ask? So you probably all have heard of DNA and proteins, and DNA sits sequestered in the nucleus of a cell, and it's copied into RNA, and then from the RNA, the cell makes proteins. And proteins are really the workhorse of the cell. They're the thing that go around and do things. They may be structural, or they may be enzymes, and facilitate various reactions. So you've probably all heard of the Genome Project. So about actually 12 years ago now, the first draft of the human genome was published, and that was a international effort to sequence all of that data. And what came out of the Genome Project is that in humans, we have about 21,000 protein coding genes in there. And actually, it's interesting to note that after 11 years or 12 years, we still actually don't know the final number of genes. But effectively, because we have I should say that from a gene, you basically make one protein. That's the base level. So we have at least 21,000 proteins, but it's actually much more complicated than that. Because most proteins actually have different versions. And this is just one example of a protein called calumenin. And this is showing that this protein actually has two different versions, and the differences are shown here. So don't worry about the colours, but you can see that the shape of this is more or less the same in lots of places. We have these spirals here and spirals here, but there's various regions where there are differences. And this is just showing two different versions of this particular protein, but there's actually many more versions of that actual protein. So we actually are going up a factor here. The number may actually be up to, in 20s or 30s or even more. But actually gets more complicated than that as well, because these different versions are actually modified in different ways by different cells. So, and this is where other small molecules are actually attached to the protein. So, and that actually is making effectively a different version of those proteins. And this is a kind of complicated looking slide, but it's actually pretty simple. So the purple or the pink squiggle here is a protein, and this is just showing that different sugars, these units, are actually attached onto the protein. And one here is, so you can see here that this blue one's put on here, and then this green one's snipped off, and then this other one, green one, snipped off. So effectively every single one of these is effectively a different molecule. You could argue that. So this is actually one version of a particular protein, but there's actually 13 variants shown here with different types of sugars that are attached to it. And you should say, well, what's the big deal about all of this? But effectively, these different versions can all have different functions within the cell, because as I said, the protein's actually the thing that goes around the cell and does things. And the different protein versions can have health implications. So this is the example I showed at the beginning of calumenin, and actually both of these particular versions here are associated with breast cancer. And there's other versions of calumenin that aren't associated with breast cancer. So the proteome effectively is the full catalogue of all proteins in an organism. And in this project, we're just talking about the human proteome, but I should point out that the software that's being developed is applicable to any other organism as well. So the full catalogue of proteins and all of those many types of variants is what constitutes the proteome. As I said, understanding that is actually very important for understanding protein function in health and disease. You know, the genome's been a great, start at that, but the function really is happening at the protein end of things. So it's pretty hard to predict how many proteins and types of or variants of proteins that there are, but based on 21,000 genes, a sort of conservative estimate is that there's probably over 2 million versions of proteins and their versions in humans. So, having said that, actually only a small proportion of the proteome has been experimentally detected. So detecting proteins is not as simple as sequencing DNA, and it's performed by other methods such as mass spectroscopy, generating antibodies against these proteins, and it's just technically, I guess, more difficult to do on a high throughput scale. However, the Human Proteome Organisation is committed to identifying all proteins and their variants. So HUPO is an international organisation. It's the Human Proteome Organisation. They effectively, as I said, they want to identify all proteins and their variants, and they have two sort of broad approaches to doing this. One is based on disease, and I'm not going to talk about that today. But the other one is based on information we already know from sequencing the genome, and this is the Chromosome-Centric Human Proteome Project. So it's predominantly a nationally organised global effort. So the reason why they're going down this chromosome approach is that it provides an information basis for what proteins should be there. If we know that there's a gene there that we think produces a protein, well, it's a pretty good indication that, hey, there probably is a protein. Let's go try and find that protein and find some evidence for that protein. So there was formation of this has been going on for a long time, but the official kickoff meeting was just last month in Boston. And you can see here that, as I said, most of these chromosomes have been assigned to single countries. Some are joint countries here. And you'll see here that Australia and New Zealand are responsible for chromosome 7. So just back to this particular project we're talking about here, the Proteome browser. The goal of it is to produce a data integration and analysis software system primarily for the Australian chromosome-centric human proteome effort, and to make data collections from this resource discoverable through ANZ's Research Data Australia, which I'm sure you've all heard of, but it's our discovery service for research data collections within Australia. And I guess I should just point out, and I think Aneeth is going to talk about this at the end, is that even though the project is being developed for chromosome 7, there's widespread interest in this consortium. So there already exists many databases that hold proteomics information, and these are just some examples here. Some of these are also genomic information, but they also hold some protein information. But NextProt is an example of a database that's it's curated. It's already an aggregator of information from multiple data sources, such as SwissProt, if you've heard of that. GPM, same deal. It's a sort of global community effort that is aggregating data about proteins. GlycoSuite has information on the glycosylation, which is the attachment of those sugars. PRIDE and the Protein Data Bank hold structure information. And I guess the others here, what specifically we want to talk about is Peptide Atlas, which is developed out of Seattle, and the Human Protein Atlas, which is a Swedish-based project, and they are developing or generating antibodies towards every human protein and looking at where these proteins are expressed in various tissues. So really what was lacking in the effort, or what was lacking in this chromosome approach, was an integration of all this information that already existed. There was no sort of overriding integration and no easy way to digest that information. So this project, 6 months, it's been going for 6 months-ish. It's got 8 months, okay. It's got another few months to go. This is an overview of the version, the first software version that's been released. And effectively, I don't know if you can see this at the back, but what it allows someone to do is just select a chromosome, hit this button, and then it's basically showing all of the genes here in order along that chromosome. And then all of these rows here are presenting evidence of is there a protein that's expressed from that gene. The colours here represent evidence. So there's various, I guess, confidence levels of whether the information is good or whether it's inferred and things like that. So basically green is good, yellow is that there's some evidence, and red is that the evidence is not particularly great, and black is that there's no evidence whatsoever. So each of these rows here can be expanded, and so the first one here we have mass spec data, various different rows here. This is antibody information here, and then there's other. So if someone comes along and clicks on a square here, it actually expands that. So in this particular protein, whatever it is, I can't actually read it, but they'll say that there's actually the sources. So NextProt says that this has reasonable information, and GPM says this has very good information. And then we can go there, go into this, and then actually go to the original data that was in the database, the other database. So effectively, what this tool is allowing is a global overview for this chromosome-centric protein atlas effort. The information here along this y-axis is the integration of data from various resources, and the information on the x-axis here is, well, at the moment it's simply based on chromosome, but effectively any information that is already compiled about genes, we can actually compile the information on this axis about that. So at the moment it's chromosome number, so it's just showing us what's there on chromosome 7. But what's really useful for this effort is to say, well, actually just show us the ones where there's no data, and we just want to focus on those. Or you may want to say, just show me things in a particular gene family, or a protein function, so just show me transcription factors across the whole genome. Show me proteins that may be associated, or where the gene is associated with a particular type of disease, and cellular location. So those types of, I guess, collections that can be generated through dividing or looking at different genes with various specific associations are going to be published into Research Data Australia. So these are examples of, well, actually, what are the known proteins associated with a genetic location or a particular disease? Maybe all the proteins that are associated with the cell membrane, ones that are associated in a biological process or in a molecular function. So these are obviously, if you know anything about this, is going to use the gene ontology and other gene-centric existing evidence-based methods for classifying genes. And there's potentially hundreds of thousands of collection descriptions that will be published into RDA, but the reason for doing this is that it makes the tool discoverable. Because maybe someone's specifically looking for proteins involved in biological process. So now I'm going to pass over to Anita.
Thanks, Jeff. So Jeff's given you a context about the research context to this project. The proteomics researchers worldwide use something called a transproteomic pipeline. What that involves is they actually put in their raw data into worldwide databases like Tranche and Pride. Now from Tranche and Pride, it gets picked up and curated by sources like NextProt and GPM that Jeff showed you on a previous slide. Now, there are other data sources that we are collating as part of this project. So some of the data sources down here would be GPM, NextProt, Human Peptide Atlas, Human Protein Atlas, and Peptide Atlas. What we have, the system's architected to be extensible. We've got cron jobs, basically batch jobs that run periodically. At the moment, they are configured to run weekly to pull these data sources and see if there's new data that can be pulled in. we are pulling in data from NextProt and GPM into our database at the moment, and data from Ensemble, which gives us the entire gene sequence. So that's our baseline gene data. We have a process that runs overnight that actually analyzes this data and creates the traffic light. The traffic light that you saw on a previous slide, if you had to generate this on the fly, it could take a lot of compute. We've done that, so performance is really good on the website. People can drill through and drill down into many layers of data that has been pre-analyzed, and then drill through out to the original data in GPM, HPA, Peptide Atlas. We have metadata, and the collections that Jeff mentioned are metadata, standard metadata that can be pulled out of from the analysis and put onto our OAI PMH data provider, which gets harvested by ants. At the moment, the web interface, I think, is the ideal interface for researchers because it allows them to see it on a single web page. You can actually get a holistic view of all the proteomics data that's available for a chromosome or a specific gene set. At the moment, we started off this project earlier this year, focusing on the researchers in Australia. We have been talking to proteomics researchers throughout the country, and we've got their feedback, and based on their feedback, we developed the system incrementally. When this was presented, the concept was presented at Beijing, and we got very good feedback from the Koreans and the US teams. We've had a lot of interest being expressed by the Americans. The Americans and Koreans are basically behind us all the way because they see this as as a useful tool for them, and there's no point in reinventing the wheel for each of the countries that are involved with this project. At the moment, in fact, today, there is a meeting of the bioinformaticians from all of the international teams are having a discussion. And at the moment, the Russians have another tool out there, but we're hoping that we can consolidate these efforts and create a single tool that can bring together all the efforts and support the entire CHPP project. Ian Smith is the chair of us, is behind this entire project at Monash. He sees, earlier this year, there was a wonderful vision that was put together to see this project supporting the CHPP effort, not just in Australia, but internationally. And it's hoping that this will further the proteomics researchers and help them get to their research outcomes. Just left for me to acknowledge the effort that has gone in from not just Monash Research, but the HUPO steering committee and the leaders of this entire HUPO initiative, Bill Hancock and Yonki Pike, and all of the data sources that are behind this initiative. Thank you.
Thank you, Jeff and Anita. If you have any questions, please come to the microphone and introduce yourself and your affiliation.
Hi, Ed Witsky, University of Cape Town. Is this all accessible via the cloud, or do you require a dedicated server at your institution?
No, it's a web application that we are hosting. So anyone with web access should be able to get to the system.
Hi, Jason Swedler from University of Dundee. So it seems like the presentation of the data takes inspiration from the sort of gene tracks that Santa Cruz, UCSC, et cetera, have, and that's great because that's such a great standard and it works so well. But thinking about the data that you're going to be generating, so I mean, our lab is generating analysis in HeLa cells and someone else in U2S, and already there are differences in P53 right there, right? So how do we scale this? How does that, and so I'm less concerned about the data because I'm sure you can, but the presentation really of the complexity of that and post-translational modifications, et cetera. So where does that all go?
There are two parts to this question, I think. The first part is you're talking about experimental data that labs might have that isn't yet published.
Well, so when you talk about a chromosomal proteome, chromosome 7 in what background? And so I presume, so chromosome 7 in fibroblasts is different than chromosome 7 in endothelial cells, et cetera. So there's that richness, and then there's the cell lines, and the data sources that you're pulling in have all of that diversity as well. So
I think the original idea of the matrix is actually based on, well, partly it was based on things like Chromosome-Centrus, but also looking at hierarchical clustering matrices, where actually the dots are very, very small. And actually what, in some version of this in the future, we'd like to be able to hierarchically cluster based on this axis or that axis. So actually you can, in theory, yes, you're right, you'll have, thousands of tracks, presumably, but you can expand it out, cluster based on, whether there's data or not data or whatever specific types of data on that axis, and then explore it that way. So actually this is actually a very, very coarse visualization at the moment.
That's good. It's cool.
But it will support more tracks being added, and we will be doing that for the next 4 months. And based on feedback, we will be it's open for discussion, and that's why I think making it available as an open source project and inviting the other teams to develop more tracks is the way to go.
Any further questions?